Dr. Rustum Karanjia is an attending physician at the Ottawa Hospital and the Ottawa Eye Institute in Neuro-Ophthalmology. He is cross-appointed to the Ottawa Hospital Research Institute as a clinician scientist. His clinical and research interests include optic neuropathies, including LHON. While Dr. Karanjia is now in Ottawa, Canada, many of you who are patients of Dr. Sadoun may recognize him from his time collaborating with Dr. Sadoun in LA, where he continues to practice part-time. Dr. Karanjia?
So thank you, Lisa, for having me come today, and it's nice to see some familiar faces in the crowd. Part of what I was asked to do today was to give you a sense as to what actually goes on behind the scenes when it comes to research, and also to try and decipher some of the language we use. When we get into medicine, one of the first things that we have to do is we have to learn a new language, and there's a couple of reasons for that. One of them is we want to be very specific. When we say something, we want to mean exactly what that is. So we have words that we've taken and made up. We do that a lot. And we've also got words that we've repurposed to specify certain things. And so we're going to try and cover a few of those things, and then Dr. Sadoun is going to go over some of those studies that we're doing now. And just as for full disclosure, I have worked as a consultant for Stealth Biotherapeutics, and I have been involved in some of the clinical trials that have been taking place in Los Angeles. So what's the goal? What's the reason why we're doing this research? Well, essentially, we want to make sure that whatever we use to treat patients is safe, it works, which is the main thing, and that patients are able to access it. And it's got to be accessible not only from physical presence to be accessible, but it also has to be accessible from a financial perspective as well. Now, this overall process is extremely regulated, as you can imagine. It's important that that regulation is in place because we don't want to put people at risk. That's one of the main overlying things whenever we proceed through clinical studies is we want to make sure that whatever we do is safe. We also have to take into consideration the cost, the length, and regardless of how good we think we are, a lot of what happens in clinical studies ends up being trial and error. You have to try something to really know whether or not it works. And so some things don't work, but we learn from it, and that's also important. So there's several agencies that are involved in regulating what we do. In the United States, it's the FDA. In Europe, the EMA. And in other countries, such as in Canada, we have Health Canada and TGA in Australia. All of these organizations are independent. So this has a few implications. If an agent, such as Idibinone at present, is approved by one of these agencies, it doesn't instantaneously mean it's approved across all other places. So each one of these have their own regulatory frameworks. And in some cases, some medications actually don't get approved in some locations, but are approved in others. Now, research is an expensive proposition, and we're fortunate in Labors to have some very good organizations to help support us and back us up. The UMDF and the LHON project is essential for a lot of the things that we do. And there are other nonprofit organizations, Foundation for Fighting Blindness and others that help as well. In addition, when you get into some of the bigger trials and studies, you start looking at some of the government agencies, such as the NIH and NEI. But when you get into the realm of clinical trials, these are extremely expensive propositions, and usually they involve some sort of industry sponsorship. It's very difficult to get a government or an organization to come up with the several $1 million associated with being able to run one of these studies independently. A lot of the research that we do is all based upon things that have come out of laboratories. And a lot of these laboratories have been based in academic centers. Some of them are based in biotechnology and pharmaceutical companies, but together, they're the ones that come up with the ideas. Once they've come up with the ideas, we have to start seeing and figuring out whether or not these ideas are going to have implications for the patients or if they're going to work. And then you get into the realm of doing some preclinical testing. Sometimes this gets referred to as phase 0. The terminology gets a little confusing at this point, but this essentially looks at cell cultures, animal models, and other things where we're trying to see whether or not this new agent actually has a working ability to do something in a live organism. We then move on to clinical research, and there's something called an IND, which is an investigative new drug, and that's an application that gets submitted to the FDA to allow patients to be tested with a new novel molecule. Now, research doesn't stop once the drug is approved. Any drug that gets approved is involved in post-market research. And so part of the reason for doing that is if you can imagine if you enrolled 100 people inside a clinical trial, When you get this drug out, you may end up having 10 times that number of patients on there. So that 100 people may not represent the entire population. As a result, you want to make sure that once you've rolled this medication out and you're treating many more people who are much more diverse than your original group from the clinical trial, you're not causing them any harm. So post-marketing research is very important. So there are two different types of ways of looking at clinical trials. There are observational studies, and John Guy and the University of Miami did a very nice observational study looking at the natural history of labors a few years ago. And those studies don't involve changing anything. We don't give you anything, we don't do anything, but we want to see how the disease progresses and how things change. Dr. Sadun's been involved in a similar study down in Brazil as well, which you may have heard of. Then we get into the realm of interventional trials. And these are trials where we take a drug or we take a therapy and we apply it to a specific disease. And then we start looking to see what happens. There's always a backup of basic science data, animal data, before we get even close to humans to tell us that we predict this is what's going to happen. But we don't know until we try it. So there are a lot of terms that get thrown around when you're talking about clinical trials. You can talk about a placebo-controlled trial, which we'll get into in a few minutes. Also randomization. But then there's also masking. And what masking basically means is that myself as an investigator does not know who has received the therapy, and the individual receiving the therapy does not know who has received the therapy if there is an arm associated with a placebo. Now, some of the trials that we'll talk about a little later on, our placebo is in one eye and the treatment is in another. So in that case, I don't know which eye got the placebo and I don't know which eye got the therapy, and the patient doesn't either. We also, in that situation, have different arms. So you can have a placebo arm, you can have a treatment arm, but even within that treatment arm, you can have different doses. So you can have multiple arms in one study. And each study has its own criteria, which tells us whether or not a patient can be enrolled. And these are set out for particular reasons. One is that you want to select patients who you think are going to benefit the most. So you want to decrease the risk to benefit ratio. In addition, you want to have specific exclusion criteria so that you don't want to enroll individuals where you think that this medication is going to be harmful. So that's why we come up with these inclusion and exclusion criteria. So it's not that we're trying to cherry pick things to make things look better. It's that we want to be safe, and we want to make sure that what we do is safe. The other thing that comes along quite often is the idea of significance. And what does that mean? Well, essentially, what you have there is a p-value. And what we refer to a p-value is the chance that this could have happened just randomly. So we set our p-values at less than 5%. And that basically means that there's less than a 5% chance that what we found in the study was due to random chance. So when we actually talk about clinical trials, there are multiple phases, as most of you have probably heard. Phase 0, like I was mentioning, is usually the preclinical work, typically taking place in animal and cell models. It also sometimes refers to the studies that get done on normal individuals looking at the pharmacokinetics, pharmacodynamics, which is basically how the drug gets absorbed, how the drug gets processed, and how the drug gets excreted from the body. Sometimes that gets bundled into phase 1 trials, and phase 1 serves to try and figure out how high you can go on the dose before you run into problems with toxicity. So it's a dose escalation where basically you start off with a smaller dose. If nobody has toxicity, you go to a higher dose, and you keep going until you reach your maximum. It doesn't look at efficacy, so it doesn't look to see whether or not the treatment works. In order to see whether or not the treatment works, you start moving on to a phase 2 trial. In a phase 2 trial, you're using that maximum dose you figured out in your phase 1 trial, and then you're trying to see whether or not you're getting an effect from that dosage. Then, if that works, you move on to a phase 3 study. And phase 3 is typically where we start enrolling placebos and randomization and other things like that. Some instances, you'll combine a phase 2 and a phase 3 study, especially in situations where you're dealing with rare diseases, where enrollment is going to be an issue, and you don't want to use up the patients that are interested by doing a phase 2 study, because sometimes those individuals may not qualify for a phase 3 study later on. And as I mentioned earlier, you always want to make sure you're safe, even after that drug is approved. So phase 4 trials are essentially, once it's marketed, looking at the medication, making sure that it's still safe once you open up the access to a broader group of individuals. So this is kind of the pathway that drugs go through. You start off with thousands, up to 10,000 compounds just to end up with one drug. So through that process, it takes a long time. You have a lot of years, up to half a decade in preclinical studies. Then clinical trials themselves can take a decade to go through. And eventually you hope, and the drug companies hope, that they end up with a new medication, and we hope that we end up with a new therapy to be able to treat our patients. But for one drug, you're looking at screening and going through this with about 10,000 different compounds. So why is LHON become so interesting when it comes to these clinical trials? Well, there are a few reasons. One is that LHON, what we learn from LHON can be potentially expanded to other diseases relating to mitochondrial diseases. And other mitochondrial diseases, what we learn from there is getting applied to LHON, so it goes both ways. So one of the interesting things, though, with LHON is that in about 95% of the individuals, they have one of those 3 primary mutations you've all heard about. And because of that, it makes diagnostic criteria a lot easier. When you get into some of the other mitochondrial diseases, the genetics are not as well worked out. And as a result, only up to about 40% of those cases will necessarily have a genetic diagnosis. So that makes it a lot more difficult when you're dealing with those mitochondrial diseases to have a very homogeneous group of individuals that all carry the same sort of disease and you're testing the same against the same. In addition, with Leber's, most patients are only affected in one system. Their eyes are affected. Whereas in most of the other mitochondrial diseases, there are more systems that are typically involved. And the other nice thing about having one system is that with that one system, the eye, we have very good ways of testing it and being able to look at it because of where it is structurally located and the access we have and the technology that's been developed. We have good tests that we can use to look at those things. Now, when you're dealing with rare diseases and these clinical trials, you typically have a numbers issue. So if you look at all rare disease trials that were run in the United States back in 2014, you'll find that the vast majority of them, almost 1,000 of them, which is pretty well 2/3 of the cases, only had up to 50 patients in it. So there is an issue of getting sufficient numbers to make sure that whatever you're doing is actually correct and that your randomization and everything else works. One of the challenges with Leber's is that individuals who haven't got the family history and the background in the disease are likely to take a lot longer to get to diagnosis than those individuals who have that family background and know about the disease. So that has implications for the clinical trials because some of the clinical trials specify that you can only enroll if you have been diagnosed within the last 6 months. And in some individuals, it's a year before they've actually been diagnosed. So that can be a bit of a challenge. And it's something that we try to increase awareness about and try to educate other physicians about so that they appreciate that it is important to try and get to these diagnoses sooner. So why do we use control groups? Well, control groups are important because we want to know whether what we're doing is not any different from the natural history. If the natural history of the disease, people get better spontaneously, how do we know that what we see from treatment is not just a spontaneous change? So what we're really asking here is, was it really the drug that made the difference, and was it really the drug that worked? So there are a few different types of controls. You can have external controls, those are natural history studies, and you can have internal controls, and these are studies which involve giving some patients no treatment or doing a dose response curve where basically as you escalate the dose you see more of an effect, or you can use a placebo. The placebo effect is actually well documented, and basically if you look at a study that was done with vitamin C, and they told patients you're going to take vitamin C or you're going to take a placebo, but patients could figure out which one was the placebo and which one was the vitamin C. Those that figured it out were more likely to say if they were on the vitamin C that they had less colds and they felt better, whereas those who figured out they were on the placebo were more likely to drop out of the study altogether because they figured it wasn't doing them any benefit. So the placebo effect runs two ways, and it's important that we maintain the masking that we do so that we don't stop biasing ourselves unconsciously. So when we get into picking patients and moving patients around and deciding what treatment, which eye gets injected, which eye gets drops, a lot of this has to do with randomization. And one of the main reasons why we do randomization is that we just want to eliminate the possibility that there's something out there that we don't know about that's impacting this disease. And we want to distribute people randomly so that the chance of that impacting one group versus the other is minimized. So that's trying to decrease the imbalance of chance alone. If you don't randomize, you also ultimately end up with a selection bias, because if I know that this patient is more likely to improve, I'm probably going to put them inside the trial phase that has the drug therapy because I want my results to be a little bit better. I'm biasing the study. I'm causing a situation where it may not actually be a valid study in the end, and it may not actually be applicable to everybody. Then you get into the history of historical controls, and one of the big challenges with that is a lot of the historical controls, if they're done retrospectively, so if we take a time point now and go back through our charts and try and figure out what these patients were like, not all of the tests that wouldn't have necessarily been done as part of the clinical trial will be done. So in that situation, we have a lot of missing data. We don't have a full layout of exactly what was going on. Patients may not have been seen as frequently as they would have been if they were part of a clinical study. So you've got a lot of issues there. You also have a time issue because technology moves along, our testing abilities move along, so you may not be comparing apples to apples. So randomization tends to produce these comparable groups that we can say that one group is comparable to the other. So whatever therapy we use is actually impacting the two groups. Randomization is also the only way that we can really end up with a valid statistical test. So the only way you get a p-value that is truly valid is if you do use randomization. Now, when it comes to the labor studies, as some of you know, we are using one eye versus the other. So you can do this control situation internally within the same patient. And this avoids the placebo arm and has the benefit that everybody is actually receiving therapy. Sometimes the drugs go through all of this and they reach a point where the data doesn't support them being adopted. And this happened in one situation with idebenone. The data did not support the use of idebenone from the randomized clinical trial. It was suggestive that it would be beneficial to patients. And if you now look at the more expanded data, it really is very strongly suggestive, so much so that the European agency actually did approve the medication. But that expanded data came from a couple of places. One of them was a named patient program where basically the patient was identified by a physician. The drug company became involved and started to provide that information to, provide the medication to the patient, and in exchange they would collect additional data to support what they had with their clinical trial. And in addition to that, sometimes even after the drug is approved, it may not be approved in all jurisdictions. You also have some governmental and regulatory issues around that, but price becomes a challenge in some situations, and I know there was some concern about that with idebenone. And also getting it through the insurance boards to get them to pay for the medication also takes time. So it's not a fast process that the FDA says the drug is approved on Monday and you can start using it on Tuesday. The other thing that we just wanted to address was the issue of patient-sponsored clinical trials. And essentially, these are studies where individuals are asked to come in, get evaluated, and then they pay a sum of money, sometimes in the several tens of thousands of dollars, to participate in a clinical study. And these clinical studies, because you're paying, there isn't really a placebo arm. It's not really something where you can advertise that you pay me $10,000 and you may end up on placebo. So there's usually no blinding, there's no masking, and there are other issues with these studies. So it becomes a bit of an ethical conundrum to know what to do with the data that comes out at the end. In addition, one of the big concerns with these studies is the safety aspect. And let's consider one study that was done looking at the MS literature. And so in the MS patient population, some of you may have heard about a year ago, 2 years ago, there was some studies and talk a lot about something called liberalization therapy. And essentially what that involved was opening up the veins inside the neck because it was felt that the venous drainage from the head was part of the reason why these patients were getting MS. There weren't any randomized control trials, and this is probably the only time that I know of where the Canadian government became so interested in this that they themselves funded a clinical trial. Now, the US has done that several times for different diseases, but it's pretty well unheard of in Canada. And so they funded a clinical trial looking at this. And the initial data set in one of the reviews that was published last year basically said that there was no evidence to support that this was having any benefit to the patients. And in addition to that, there were several severe adverse events. Now, what's a severe adverse event? That's things like stroke, blood clots inside the veins, migration of the stents that they were using, swelling on the brain, and death. So these are things that we want to make sure that we avoid, and that's part of the reason why we go through all of the rigor that we do when we're designing, managing, and participating in a clinical trial. So, any questions, I guess, would be the next step. So money, there's a small portion of the funds that actually go into the actual development or identification of that compound that you're using. But you have to bear in mind, to get that one compound, you've looked at 10,000 compounds. So there's a bigger cost there associated with that. In addition, just running a clinical trial, for every one of the tests that you do, there's a cost associated with that. There's a cost associated bringing those patients in to see you, travel expenses, those sorts of things. And those are what rack up the bills pretty quickly.
So are you saying the money is mostly for travel?
No. Most of that money is going for doing those tests that you do when you come in. Because those tests aren't covered by your insurance. If you're part of a clinical trial, the cost of each one of those tests, each one of those blood tests, each one of those visual fields, everything adds up. And all of that plays into that total. And just one last note, this presentation actually wouldn't have happened without LISA, so the title really should be this.